So firstly, thank you to Alison and to the committee for asking me to present today. What I'm here today is basically a shout out to ARMS to take research on. So I want to present from what we do on the routine side and also how we're dipping our toes, is all I'll say, compared to Professor Madrigal's tour de force. We're dipping our toes into the research field, but it's a call to arms basically to come and join us. So firstly, who are we and what we do? We are a kingdom within a kingdom. We've infiltrated the IBTS since 2006 when we moved in. We got our tissue license in 2008, and as Paul alluded to, we recently got JC accreditation for the whole transplant program for the clinical side. We are the national adult program, so we have referrals from 32 counties for transplantation. And we also are the collection site for the Irish Unrelated Registry, so we collect cells for export both to the paediatric program and internationally as well. And we have a very large autologous transplantation program, so we have autologous PBSC collection and infusion. We process for the Dublin sites, so basically with the exclusion of St. Vincent's, we have patients coming in from Tala, Mater, Beaumont, including Drogheda and Blanchardstown. And we also have referrals from around the country. And as Larry has talked about, we've now also become the National Adult CAR-T program as well at the site. So basically, I'm going to very quickly go through what we actually do on a day-to-day basis. From the point of view of allogeneic transplantation, Prof. Madrigal has spoken very eloquently, and I know Diarmaid's going to follow up in terms of the registry. So ideally what we want is a sibling match. In the situation where that's not available, we will go to the registry. So the team, Richard and the team at the registry, will find us a donor and find and organise the logistics around sourcing that donor. We would, as a standard, go to the European sites, particularly to Germany, the Anthony Nolan and the NMDP program in the US. Ideally, we want CMV matched, and ideally for our lab, we also want ABO matches for transplantation. So we have two options. We can go directly to the bone marrow and harvest a bone marrow in theatre, and this gives us a pool of cells, including the stem cells, or we can isolate the peripheral blood stem cells. So we can encourage the stem cells out of the bone marrow using GCSF. The donors are given 5 days of GCSF subcutaneously, and they're collected in the same fashion with apheresis. But essentially, we end up with a blood product for processing. So as I said, with the allogeneic program, while the patient, while the patient is having their treatment, their chemotherapy or total body irradiation, at the same time, either a sibling donor at St. James or an international donor is having their collection. And the cells are collected and then transported to St. James's. That sounds very easy. We've had to deal with lots of logistics. We thought ash clouds and the beast from the east was bad, and then we hit COVID. So getting cells to Ireland in times of trouble has been fun. But we've learnt. We've learnt a lot about logistics and transportation in and out of the country. And then in terms of the cell preparation, depending on where our cells are coming from and also on the donor age and size, we may have a very good product, we may have a poor product, or we may have an exceptional product from some of our German colleagues where we have an excess of cells. We're processing and we cryopreserve. So we have a GMP facility, so obviously our main task is to ensure sterility of the process that we do. We take blood cultures at all stages of the processing. We monitor our cells while we're processing, and we're grateful to our IBTS colleagues in the EM lab who provide us weekly confirmation of our GMP status. We then assess the product. We do weight by volume, we do CD3 and CD34 enumeration to see what's in the bag, and then we decide on the dose. We are in touch with the consultant, and they decide what dose they want to give on any day to whichever particular patient it is. We also do functional assays. We do progenitor assays, so we're growing the cells in a culture media. If the cells grow in culture, they'll grow in the patient. So it is a very reliable QA because it's a 14-day culture. We release the products fresh, so we aim to harvest the donor and have the cells into the patient within 24 hours. Our expiry time is about 48 hours, and we've been put pin of our collar on some occasions. But ideally, we want to issue the product fresh, and eggs being eggs, we occasionally will have an issue. Donor ranges and patient ranges push things outside our standard specifications. So we like to release. We like to release on the right criteria, but we occasionally have to release under medical concession. We're not in a situation where it's a blood pack and we can start again. This is the only product for this donor, or for this patient, ideally, and we have to give as best we can. So, as I said, sometimes we have enough cells to cryopreserve, and also the same situation in the autologous setting, where we're getting the cells directly from the patient, and we freeze the cells in albumin, or plasma, collected at the same time as the product. And we use control rate freezer. We bring the cells down to -180 at 1 degree per minute to make sure that the cells are secure both at the time of freezing and also at the time of thawing. And we use vapor phase storage. So we really go from bench to bedside. We take the cells from the cryopreserved stock. It's transported to the ward, and the scientific teams, the medical scientists, you will have seen them trundling out the building with this shipper. They're going to the patient's bedside, and they will thaw the bag, doing full check back with the nursing staff, and it's administered. So we, as standard, would produce or process about 100 or 115 autologous PBSC products per year. And we are, as standard, doing about 100 autologous transplants per year. That's slowed down slightly because of the elective nature during COVID, but we're catching up. And then with the allogeneic products, ideally we want to give fresh, as I said, but we have had to, with COVID as well, due to logistics, and to ensure that the patient or the donor didn't get COVID during the time between the patient starting chemotherapy and getting ready. So we cryopreserved electively 46 products during the early phases of COVID. We saw no change in graft, but we did see slower neutrophil engraftment. And also, Chris Armstrong presented a poster at EBMT last year and showing a worrying increase in rate of relapse within the first year. So it's an elective procedure. We wouldn't electively cryopreserve an allogeneic product. If the donor is not, or if the patient's not ready for it, we will do. So as the previous speakers have shown, we've basically been doing cellular therapy since 1984. And as Paul alluded to, Prof. McCann inspired a lot of us in this room to stamp out leukemia, as he used to say. And we've reached a goal mark of around 3,300 transplants this year. DLI as well has been used post-transplantation to tip the balance in the immune system from what you want is a tiny bit of graft versus host disease and a graft versus leukemia, but obviously not too much. So we cryopreserve donor excess cells in graded doses. So we're giving back a very small volume. It could be as low as 1 ml of cells. And we freeze those cells in aliquots so that in the situation that the donor is showing signs of relapse, that we would intervene with this immunotherapy. So we've been doing that since the early 90s. So that is essentially what we do on the routine side of things, and until last year. Well, previous year, I suppose, in terms of standard procedures and getting things validated and organized. We became the National Adult CAR T program, and as Larry has spoken about. So we're using the experience in terms of the collection of the patient, the cell handling and processing, and also in terms of the clinical experience of dealing with graft versus host disease made us ideal for CAR T therapy. We've learned a lot, as I said, about the logistics of shipment and storage. So what we are doing at the moment is that we have, as Larry talks about, we have two commercial companies that we've onboarded with. We've worked with Novartis since this time last year, and Kite then during this year. We had to change our license to include the collection of stem cells. They are stem cells and they're under the tissue establishment until they go into that shipper and leave the building. Then they become starting material for a GMP process. As part of that, we also had to go through known consignor status, so it was a whole new world to me in terms of Irish aviation law and being a secure site to where we're putting these products into a shipper that will not be X-rayed. So we've airside clearance with the DAA is another bonus. We haven't tried to use it yet at the airport. Haven't gone anywhere since COVID, so that's something. So to date, we've collected 28 patients in the last 12 months. We've collected, with the Novartis products, we're freezing it down, and we ship it frozen. With the Kite products, they come and collect on the day. So we've shipped them out, and all bar one is out there in the manufacturing site, and these have come back. And we, again, go to the bedside with them, we thaw them, and they're infused. So it's been very exciting to be part of a new therapy. We've been doing, I suppose, stem cell transplantation has been the bread and butter. It becomes very routine. It becomes what you know. So it was nice to start something new with the CAR-T program. That's now bedding in. We're kind of getting used to it. Dare I say, we're getting the hang of things. So I suppose from there it was why would we start on research when we have obviously a very busy service? So I suppose it's always been part of the ethos of the lab. The lab was founded, as Paul alluded to, by Prof. McCann. We used to be called the Development Lab in CPL, for those in the room who may be old enough to remember that. But essentially it was always about promotion of staff and in terms of attracting and recruiting good staff. So we've always dallied, shall I say, in small projects in terms of medical science with our colleagues in TUD and master's projects for Coleraine as well. So things aligned. Alison started in IBTS and showed great support, and Steve as well, in terms of a collaboration. We also had some commercial funding which came in as well. Obviously there's a huge interest in CAR-T, and also St. James's are promoting. So there was a new research office at both the IBTS and at St. James's. So it was a perfect time, and we had a candidate who wanted to do a PhD project, Hayley, who you have heard about already. So we established a cellular therapy research group, and we've decided first of all to look at the immune reconstitution post-transplant. So this area is when the patient has their radiation and chemotherapy, their cells drop down into their boots, and they're very neutropenic during this phase. And this is obviously the phase when we are hugely dependent on the large, large donations or large volume of donations in terms of these patients are being supported by blood and platelets. In that early phase, then we have de novo generation. The NK cells are the first cells back. And then the T cells and the B cells will come back as late as day 100. So it's during this phase that the patients are very immunosuppressed, and we wanted to look at this in a little bit more detail. And we've linked up with Derek Doherty and put a plan together. So we put in a plan to ethics, and what we were doing is basically combining what we're currently doing with a new plan. So this is very much still at the very early stages, but it's more to talk about the whole evolution of getting to do some research. So we're going to take what we do as standard in green, so donor selection and HLA identification with Richard's team in using next-gen sequencing. And we're also going to look at KIR genotype in terms of is that impacting on our immune system post-transplant. We have set up, we're going to look in detail at the graft in terms of a profile. We're going to look at defined time points, and we've established a biobanking patients post-transplant, and as I'll come on to post-CAR. We're looking in detail at the immune profiling and immune functionality, and we're linking that with chimerism and minimal residual disease, which is already in place. We're going to look at the responders versus non-responders, and we are really lucky in that we have an MD, Chris Armstrong, who has been instrumental in setting up the biobank with us. He's very persuasive. He's consented the majority of patients who've been through the place in the last year. And he's already started a detailed review on outcomes using the EASIC scores and comorbidity index, and we're going to look at multi-parameter outcome analysis. As I said, the biobank seems very simple. Patient comes in, we take samples, but it's huge logistics in terms of making sure that the sample is taken on the right day at the right time. We are banking serum and plasma and cryopreserved PBMCs isolated from the peripheral blood, and the Royal We, it's a team behind getting all of these samples and getting it done. So to date, we wrote to ethics and we asked for, our plan was essentially to do 80 transplant patients, and we're already well on the way within a year of collecting that number. So as you can see, we're at 30 patients consented. And these are a group of myeloid patients, so we're working with Nina and Chris, and we've identified a specific patient cohort getting a specific treatment. They're getting reduced intensity allogeneic transplant, and we've been following them through transplant. And as you can see, some of these patients are now day 360 post-transplantation, and we have those samples banked. Haley had a poster upstairs. I'm not sure if they're still there, but we have shown and optimized with Derek's help immune profiles, and we're looking at the T cells, the B cells, the innate cells, and identifying those very rare events, and we hope to correlate that to the clinical outcome. So having set that up in the stem cell transplant, obviously we wanted to use the same profiling in the CAR T setting. So again, we're going to look at the patient in terms of their profile. We're going to run their cure profile. We don't know if that has any impact on the outcome in the immunosuppressed state. There is very little on that, so we thought we would do that. We are going to do detailed monitoring of the product that we start with. So the MNCA, the mononuclear cell apheresis, is the autologous apheresis product that goes out to Kite and Novartis for manufacturing. And then we're going to look at the patients post-transplant. So again, we're looking at immune profiling, functionality, and we have the samples. I'd love to move on and do some omics. What they are, I'm not exactly sure, but you know, if we have more funding, we would be able to do more. So we are going to again look at patients' response. In terms of the responders and the non-responders, and those who do respond, those that hold response. And we're also going to be looking at the CAR T, identifying the CAR T in the patient and whether they retain that and if that's any impact. And again, detailed medical outcome and analysis. As I said, Chris has persuaded 16 of the 27 patients going through the system to join the biobank, and they've been very willing. And again, we've been monitoring those patients as they go through and collecting those samples and banking them. So I suppose one of the things around the CAR T is that it's obviously of huge interest, and not only for the routine and the clinical side of things, but also for the pharmaceutical industry. Obviously, it's a huge interest. And I suppose the pharmaceutical industry in Ireland is a huge and important industry for Ireland Inc. And NIBRT and have set up a cellular therapy forum, and we met Avectis at that forum, and we organised a research agreement with them to, again, as I said, very much a preliminary study on looking at their technology, which was the Cellular technology for transfection in CAR T construction. We went for ethics, and I'll shout out to Anna and Erin, and again, the poster was presented here. So we actually, because of COVID, this was unfortunately timed right into 2021, 19 and 21, and the Avectis equipment that we wanted to use, the clinical use, was not available to us. It wasn't through yet. So they very kindly agreed to proceed with the project and gift us funding. So we've used a different transfection device. We used a BTX that was available, thankfully, within TTMI. and we electroporated the cells. So basically the electroporation is shocking the cells and allowing the cells to take up the package, if you like. It's a very versatile and inexpensive technology. It is gentler, or they reckon it's gentler, and will result in a more functional T cell, possibly. The only, I suppose, the thing around it is optimization of the process. So I won't go through this all in detail, but again, it's very preliminary stuff. We identified normal donor buffy coats, Alison, used as normal donor buffy coat controls. We've cryopreserved them. We used patient cells from patients who were no longer potential candidates. And we used green fluorescent protein mRNA. Again, we would love to have used CAR mRNA. It's extremely expensive. And again, because it was COVID, every mRNA facility in the world was making mRNA for vaccines. So GFP protein was available to us, and we used it. So we used the electroporator, and we got actually quite poor electroporation. We were down in the single digits, as you can see here. We were expecting it to be a lot higher. So again, small fry. We looked at myeloma and lymphoma. Our lymphoma patient was substantially better than our myeloma patients, but again, it's very small numbers. One thing that was of interest, and we were looking with Derek in terms of looking at the different cells that were looking at the expression in the different subsets to see was there a difference between the different types of cells. So the culture is a 3-day T cell culture, so not surprisingly, the percentage of CD4 and CD8 cells is high. And then you have very low levels of the innate cells, so MATE, INK, and NK cells. But when we did the electroporation, the CD4 cells took up the GFP expression, as you can see, very highly, as we would have expected. The CD8s, 50-50, not so much. But interestingly enough, the rarer cells really liked it and took up the GFP protein. So again, this is all very new, and where it goes, we really don't know, but it will go somewhere. And I suppose that's the benefit of trying something and seeing where we go. Future directions, I suppose one of our key things will be to establish a biobank and make it a permanent fixture. We want to look at the immune function and profile, as I said. And the idea behind this is that we will monitor any changes to protocols that we use. So we're setting, basically we're looking at current state in terms of our protocols, and then if there's a change, do we see a change in outcome? And also to develop the monitoring in terms of CAR-T, the monitoring of it as a routine process as it evolves. As I said, with CAR-T, we've seen amazing things to come, and they will come. But I suppose one of the issues around it, as has been discussed, is the availability and accessibility of CAR-T to most people. So on-site manufacturing of CAR-Ts is something that is definitely feasible, and is possibly something that we will be looking to in the medium to short term. And then whether or not an allogeneic CAR bank is also something that's coming in the future. Personalized cell manufacturing is coming, and it's, I suppose, for the discussion is where does it lie? Does it lie within a hospital, an academic hospital? Is it within the HSE, as in, is it a remit within the blood bank? Is it a cell that should be in a tissue bank? Or should it be commercial? Or should it be all of the above interacting? So it's the way forward, but ideally we want to evolve what we have and future-proof our processing facility and our staff. And I suppose that's where the research group was. I suppose ideally we want more people to join us and think about doing more research. I suppose this week is National Science Week. I suppose everybody in the room was at some point as a child interested in science, whether it was the nature walk or a chemistry set, it set you off. If I can do it and dabble in research, I advocate for everybody to try it. There's the routine side, but there's also the buzz of finding something new and getting involved in research. So I would very much advocate to talk to your colleagues. There is always something, even if it's something small, a case study. Get involved, get talking and collaborate. So thank you very much.
Thank you very much, Nicola. We are running late, but we have time for 1 or 2 questions, please. Ole, please.
I was just wondering, I noticed in both your presentation and in Larry's, with my virology hat on, you mentioned potential viral reactivation. And I was just wondering if you expect that to be different depending on the stem cell transplant or CAR T therapy, given the fact that they're going to, I guess, interact with the immune system differently.
I'll let Larry answer that question.
Yeah, it's probably going to be very different. It's going to be very different between the allogeneic product, where we've obliterated the T cells and they don't have that immune, that potential for immune attack, I guess, on the viral cells. So far in the CAR T program, because it's an autologous product, it's very gentle lymphodepleting therapy beforehand. We're not, and we're giving back T cells, I guess. I know they're genetically engineered to hit the CAR T, but they'll have their own T cell proliferation potential. So for now, we leave patients on Valtrex for the year, just because we can. It's cheap and we can do it. But so far, we haven't had any trouble with viral reactivations in the CAR T cohort, unlike the allogeneic program. Having said that, there are two patients in the UK, one of them which we had sent over with the new product Tecartus, which is a different manipulation of T cells. So they're a different manipulation of the T cells in their starting product. 2 of those patients have died from HHV6 infection in the brain. So there may be something different to come with different products as we start again manipulating the immune system. But for now, with what we're doing, we're not seeing anything yet. But we're 1 year into the product and 25 patients, so we'll see.